Good day, everyone. It is a pleasure to share some of my work at the Sophion User Meeting 2021. I'd like to start by thanking the Sophion team for providing us with this great opportunity to showcase the work we have developed at the University of Queensland using the QPATch instrument. My name is Fernanda, and I'm a research fellow at the Institute for Molecular Bioscience at the University of Queensland. As most of us are aware, ion channels are involved in a number of complex diseases. Studies have shown specific subtypes of ion channels are underlying pathological events in several diseases, as illustrated in this schematic figure. We are interested in the involvement of sodium and calcium channels, which are proved to participate in conditions such as neuropathic pain, including visceral pain, cancer, motor neuron disease, periodic paralysis syndromes, and epilepsy. Sodium and calcium channels share similar structure that contain 4 homologous domains connected via intra and extracellular loops. The 3-dimensional structure have been determined via cryo-EM, as illustrated for the subtypes NEV 1.7 and CAP 3.1. Noting the pore is presented by a red circle. Each domain uses a different color. And for the NEV 1.7, additional auxiliary subunits are attached to some of these domains. Our laboratories are particularly interested in the discovery, identification, and pharmacological studies of modulators of sodium and calcium channels. The main instruments used to perform our work are the high-throughput screen platforms QPATch, used in the automated whole cell patch clamp studies, and the flipper utilized in fluorescence imaging cellular assays. Typically, we initiate a study by screening for bioactive molecules modulating ion channels using the flipper instrument, as represented in the left graph on the slide, where we have performed a screening of spider venoms for inhibitors of NEV 1.7 and NEV 1.6. We then characterize these isolated actives using the automated whole cell patch clamp instrument QPATch, which allows a rapid and complete characterization of our bioactive leads over complete family of ion channels. This includes subtype selectivity and mode of action studies. It is important to mention that this is possible only because of the availability of recombinant cell lines stably expressed in ion channel subtypes. As our studies move to more detailed structure-function relationship characterizations, the need for new cell lines expressing particular domains of each ion channel are needed. For example, we are very interested in understanding the binding sites of modulators to these channels. In this particular study published in 2017, we investigated the activity of the inhibitor DF1A on individual domains of NEV 1.7 in order to define its binding site. This work was developed in oocyte system, and we are now developing these studies in mammalian system in my lab. The mammalian system allows a much more precise affinity, which resembles the IC50 of the wild-type channel in mammalian. While for the oocytes experiments, a larger concentration of compound is usually needed to produce clear affinity responses. Another study that is often part of the characterization of ion channels in diseases included studies of mutated channels, such as this work from Cardoso published in 2018. They investigated a number of mutations in the NEV 1.5 channel, which are associated with the long QT and Brugada syndromes. Again, this study was possible only on a oocyte system due to the large number of NEV 1.5 mutants investigated. More critical than ever are studies of interactions between modulators and ion channels in order to define exactly which residues are participating in these chemical interactions. While we are satisfied with our in vitro data collected via automated electrophysiology in QPATch, which investigates residues in the sequence of a peptide inhibitor, the understanding of how the peptide interacts with the channel, usually performed via in silico molecular docking studies, still remains incomplete due to the lack of information of mutations in this channel binding sequences performed in vitro. So additional pharmacological studies with mutated ion channels are essential to support in silico observations. So in summary, what would we like to know more about ion channels? Current questions evolve around the property of ion channels in species used in preclinical models, for example, rat, mouse, zebrafish, dog, pig. What are the binding sites of modulators on multiple on-target and off-targets, for example, which domain 1 to 4 from sodium, calcium, and potassium channels these modulators bind? What is the effect of ion channel mutations involved in complex diseases in the actual activity of the ion channel? We also would like to introduce single and multiple mutations in ion channels in order to support the structural functional studies. There are a number of ion channel studies that depend on a considerable number of ion channels variations and mutations to support current in vitro and in vivo observations. But to achieve that, we need mammalian cell lines reasonably expressing these ion channels variations to progress with our studies. So what are the cell options available? Remembering we are now focusing on understanding the function of these channels in mammalian system as we are interested in understanding diseases that affect humans. The initial studies I showed you mostly use cell lines stably expressing recombinant ion channels. These were generated by stable transfection methods where the recombinant DNA integrates into the host genome. These are very difficult and complex to obtain, and they are laborious and time-consuming, but they are still worth it if you are going to use the cell lines frequently and in long-term studies of a particular ion channel. On the other hand, transient transfection will produce the recombinant DNA outside the host genome. It is much easier to obtain, less laborious and less time consuming, and it's ideal for short-term studies of a particular ion channel, wild type or mutated version. Based on this previous common knowledge and our experience in regards novel studies of ion channels, we decided to investigate and select transfection methods that would facilitate these studies in a short-term and rapid way. We tested 3 different transfection reagents commonly used in studies of ion channels: the classical calcium phosphate DNA precipitation method, the widely used transfection via lipofectamine, which are liposome-based transfections, and a more modern method, the fusion reagent that transfects cells by a complex of DNA with cationic polymers. With these 3 transfection methods in hand, we decided to interrogate how different cell lines would behave in regards endogenous responses for the ion channels being studied, and an important parameter, how their cell membrane would behave under the stress induced by these transfection reagents. We could not stress more how important is the membrane integrity for whole cell patch clamp studies. We also investigated how different temperatures would affect recombinant transient expression in order to obtain high levels of expression without induced cytotoxicity, as well as different incubation periods to determine the time post-transfection with best expression levels. We tested these transfection methods in 2 independent assay systems, often used in our groups, and these include the QPET system developed by Sophion. Back in 2016, our group was very delighted to receive the PhD candidate Mahadhi Hasan, in which project required a large number of ion channel mutations assayed in both QPatch and FIPA system. Mahadhi was well positioned to test these different transfection methods and rapidly apply them in studies of ion channels involved in chronic pain. So I'd like to end the first part of this talk by thanking the Sophion team for the excellent opportunity to share my work with you all, and more importantly, for the development of such fantastic instruments that have shaped for better the way we study ion channels and investigate novel modulators with therapeutic applications. I also would like to thank our funding agencies, and more importantly, the research students Mahadhi Hasan, Hu, and Saja Malawi that developed most of the work I showed in this first part of the presentation. Thank you.
Thanks a lot, Fernanda, for your great introduction. My name is Mahadhi, and from now on, I will continue for the rest of the presentation. We optimized transient transfection to express two voltage-gated ion channels, CAV 2.2 and NAV 1.7, which are important for pain modulation. CAV 2.2 and NAV 1.7 channels are multi-subunit complex. CAV 2.2 channels consist of pore-forming alpha-1, extracellular alpha-2 delta, transmembrane gamma, and intracellular beta subunit. However, for transient transfection of CAV 2.2, we used alpha-1b, alpha-2 delta-1, and beta-1 subunit because these subunits are sufficient to perform physiological function of the channel. On the other hand, NAV 1.7 channels consist of pore-forming alpha-1 and extracellular beta subunit. Again, for transient transfection of NAV 1.7, we used only alpha-1 subunit. We optimized our transfection by transiently expressing CAV 2.2 using fusing reagent. We measured the ion channel responses from transfected cells on flipper. In this process, we tried three different cell lines, HEC293T, CHOK1, and COS1 with three levels of cell confluency, 40%, 60%, and 80%. We also tested two incubation temperatures, 37 degrees Celsius and 28 degrees Celsius, and two incubation lengths, 48 hours and 72 hours. Lastly, we tried three DNA versus transfection reagent ratio, 1:2, 1:3, and 1:6. From our optimization, we found that HEC293T cells with 60% confluency gives the best result. We also found that 1:3 ratio of DNA versus reagent provides better outcome. Interestingly, we found that combination of 37 degrees Celsius and 28 degrees Celsius incubation brings higher responses from the transfected cells. In our experiments, we found that 16 hours at 37 degrees Celsius followed by an additional 56 hours at 28 degrees Celsius is the most suitable for robust transfection. These concentration response curves are showing how the response from transfected cells were different for HEC293T cells compared to CHOK1 and COS1 cells. For CHOK1 and COS1 cells, there was no significant difference between the calcium channel response from transfected and untransfected cells. However, responses from HEC293T cells were much higher for transfected cells compared to untransfected cells. In the bar graph in the bottom corner, the effect of temperature has been shown. It is clear from the graph that incubation of 16 hours at 37 degrees Celsius followed by an additional 56 hours at 28 degrees Celsius gives the biggest response from the transfected cells. We wanted to confirm if the optimized conditions are suitable for the other transfection methods like calcium phosphate and lipofectamine 3000. To verify that, we co-transfected eGFP plasmid along with the CAV 2.2 subunit plasmids using all three methods. When visualized under a fluorescent microscope, all transfected cells showing strong fluorescence, indicating successful transfection was achieved in all three methods. The transfection efficiency of the calcium phosphate, fusin, and lipofectamine 3000 mediated transfected cells measured by eGFP fluorescence intensity was 49, 58, and 51% respectively, which is more or less similar. We evaluated CAV 2.2 channel activation and inhibition by using a flipper-based assay. For CAV 2.2 channel activation, intracellular calcium increment was measured in response to increasing concentration of potassium chloride. The EC50s of potassium chloride activation from calcium phosphate fusin and lipofectamine 3000 mediated transfected cells were around 38, 36, and 39 millimolar. respectively. These potassium chloride evoked responses are fully inhibited by the omega chronotoxin C60, which is a selective blocker of CAP2.2. The IC50s of C60 inhibition of these responses were around 19, 15, and 16 nanomolar respectively. Here you can see the raw current traces from Flipper. It shows that the response over baseline for CAP2.2 activation from calcium phosphate fusion and lipofectamine 3000 mediated transfected cells were almost similar. Similar to CAP2.2, the activation and inhibition of NAV 1.7 channels were evaluated on Flipper. The NAV 1.7 channel activation intracellular calcium increment was measured in response to increasing concentration of veratridine. The EC50s of veratridine evoked activation from calcium phosphate fusion and lipofectamine 3000 mediated transfected cells were around 28, 29, and 39 micromolar respectively. These veratridine evoked responses were fully inhibited by tetrodotoxin, which is a potent NAV 1.7 blocker. The IC50s of TTX inhibition of these responses were around 98, 95, 95, and 101 nanomolar respectively. The raw current traces from Flipper here shows that a response over baseline for NAV 1.7 activation from calcium phosphate fusion and lipofectamine 3000 mediated transfected cells were almost same. The biggest challenge for us was to ensure that the transfected cells are robust enough to use in automated electrophysiology platforms like QPADs. Hence, we got good results from Flipper to confirm the high level of expression of the ion channels. We further characterized transiently expressed CAP2.2 and NAV 1.7 channels using QPADs. For the CAP2.2 assay, the average current sizes of calcium phosphate, fusin, and lipofectamine 3000 mediated transfected cells were around 330, 790, and 1010 picoampere, whereas for NAV 1.7, the average current size were 609, 1156, and 1307 picoampere respectively. In both cases, lipofectamine 3000 mediated transfected cells produce the highest amplitude of currents. Here I am showing the representative calcium and sodium currents obtained from QPADs. For CAP2.2 currents, the depolarization to Vmax from a holding potential of -100 mV were done for 120 milliseconds before and after perfusion of 20 nanomolar and 250 nanomolar C6D. And from the graph, you can see that 250 nanomolar C6D completely block the current. For NAV 1.7 current, the depolarization to Vmax from a holding potential of -80 mV and prepulse of -120 mV were done for 20 milliseconds before and after perfusion of 200 nanomolar and 1 micromolar TTX. Here also you can see that 1 micromolar TTX completely blocks the current. The current voltage relationship of CAP2.2 and NAV 1.7 channels were also determined in QPADs. The current voltage relationship of CAP2.2 showed a partial block of the peak current in the presence of 20 nanomolar C6D, and no shift of Vmax was observed in the transfected cells compared to the non-physiological properties of this channel. Similarly, the current voltage relationship of NAV 1.7 showed a partial block of the current in the presence of 200 nanomolar TTX, and no shift of Vmax was observed in these transfected cells as well. Finally, we wanted to measure the success rate of each of the transfection methods. We tested 24 cells using calcium phosphate, fusin, and lipofectamine 3000 mediated transfection, and the success rate were measured by determining the percentage of cells that successfully completed the experiment and produced reasonable CAP2.2 or NAV 1.7 currents. From the table, you can see that the fusion-mediated transfection had the best success rate as it was significantly higher compared to the lipofectamine 3000 mediated transfection method. To conclude, I want to say that we have optimized the chemical transfection method for transient expression of voltage-gated ion channels and used it in high-throughput assay platforms, including QPATH. When transfection and whole cell formation were achieved, lipofectamine 3000 produced the largest current for both CAV 2.2 and NAV 1.7 channels. In terms of success rate, the fusion-mediated transfection performed best in the QPATH assay. The bottom line is, with proper optimization and correct selection of method, chemical transfection can be used for robust functional expression of ion channels for consistent electrophysiological recordings. I would like to thank my supervisor, Professor Richard Lewis, and Dr. Fernanda for their guidance in this project. I would also like to thank everyone from Lewis Group for helping me in different way. Last but not the least, I would like to thank the organizers for arranging such a useful meeting and inviting me to showcase my research. Thank you all.